given that it coincides with both Animal Health Awareness Week and also Antimicrobial Awareness Resistance Week, which seeks to raise awareness of the importance of good animal health from both economical and sustainability perspectives. So this webinar, in line with the themes of animal health and One Health, will highlight the impact of parasitic infections on Irish farms, the current treatments, and the emergence of drug resistance parasites. We then will look to see, from an omics perspective, how we can overcome this by potentially having vaccine therapeutics as an alternative to drug treatment, identify novel vaccine therapies, novel methods in which to diagnose infections, and then also looking at it in this parasitic disease in the global context. So we have a panel of 4 speakers from our lab and collaborators. Just for this session, we will be doing a Q&A at the end of the session, not in between talks. So if you do have questions, a colleague of mine, Carolina Verissimo, will be doing the Q&A at the end, but make sure to put your questions into the Q&A, not the chat, and they'll be addressed at the end of the session. So first up is Dr. Orla Keane. She is a researcher at the Animal and Bioscience Department in Teagasc. Her research focuses on animal health with an emphasis on host pathogen interactions. An understanding of such interactions is increasingly important due to social concern regarding the use of antimicrobials, the interactions, the emergence of antibiotic and helminth resistance, the effect on climate change on pathogen epidemiology. Orla's work aims to understand the role of host and pathogen genomic variation in controlling infection outcomes and disease progression. Orla works closely with the molecular parasitology lab on vaccine trials against parasites of our parasite of interest, Fasciola, hepatica, the liver fluke in sheep. And her talk will be looking at parasite infections on Irish farms, current treatments, and the emergence of drug resistance in these parasites. So, Orla.
Thanks, Richard. So I will just... Okay, is that showing up okay?
Yep.
Yeah. Okay, great. Okay, so thanks very much. As Richard said, I'm Orla Keane and I'm a researcher based at the Animal and Bioscience Department in Teagasc. And really what I'm going to do today is just kind of give an outline of the agricultural industry in Ireland and set the scene in terms of why parasites are such a big problem in our agricultural system and why there is such a desperate need for new sustainable control measures. So I'll briefly outline the structure of the agricultural industry in Ireland and why internal parasites of ruminants are such a big problem. Then this particular session is going to focus on liver fluke. So I'll describe the liver fluke life cycle and what is current practices on farm for the prevention, diagnosis and control of liver fluke. A lot of that control is heavily dependent on the use of flukicides. So I'll just go through what classes of flukicides are out there at the moment and the emergence of resistance to these flukicides and why this means there's an absolutely urgent need for new sustainable control measures for internal parasites, including fluke. So anyone who's driven around the country will be well aware that there's a large amount of agricultural land in Ireland. There's about 4.9 million hectares. And again, driving around the country will tell you that the vast majority of the agricultural land is in grassland in Ireland. And this is because the raising of ruminants, primarily cattle, but also sheep, is a major industry in Ireland. So there's about 137,500 farms in Ireland, and the majority of them are small. They're family farms, so they're family owned and operated. And in the country, there's just over 7 million cattle and 5 million sheep. And we also have pigs and poultry. Now, agriculture has got a huge economic value in Ireland, and it is our most important indigenous industry here in Ireland. And the export of agri-food products is valued at about 14.5 billion euro per year. And they account for about 10% of our overall merchandise exports. If you look at our agricultural output, reflecting our grass-based system of production and the large number of cattle in the country, the majority of our output is from products from cattle, so effectively dairy and dairy products and beef. And the agri-food sector accounts for about 10% of employment in Ireland when you count both direct and indirect employment. And it's a particularly a major contributor to economic activity in rural areas and in areas such as the Border Midlands and western part of Ireland. Now, because we have this grass-based system of production in Ireland, that means the animals are primarily raised outdoors. They're grazing grass for long periods of the year, and this leaves them very exposed to parasites of ruminants that live on the grass and that are ingested when these animals are grazing. And there's a large number of different parasites of concern, but some of the major internal parasites would be coccidia, and these would infect young calves and young lambs. They can cause quite severe infections, but generally over time, animals will develop immunity to them in a reasonably short period of time. We also have the gastrointestinal nematodes, the likes of these worms here that live out on the pasture. There's a large number of different species that infect cattle and sheep. Again, primarily an issue in young stock, and usually by the time the animals are 1 to 2 years of age, they'll have developed some level of protective immunity. Lungworm in cattle is a major issue as well. Again, can cause very severe infection and deaths in young calves, although over time immunity can develop to lungworm as well. And then when we come to the flukes, the major flukes would be the liver fluke, Fasciola hepatica, and also the rumen fluke. And the liver fluke in particular is a parasite for which immunity does not develop readily. And so animals remain at risk of liver fluke throughout their lives. And it's quite a pathogenic parasite. In terms of the rumen fluke, it is less pathogenic. The adult rumen fluke is believed to be quite well tolerated, although the larval stages in the intestine, if there's large number of larvae, they can be quite pathogenic as well. So in terms of the impact of these internal parasite infections, well, it's been estimated that they cost the Irish agricultural industry about 234 million euro annually. And the vast majority of that would be in lost production, so in reduced meat and milk yield. But there is about 40 million euros spent annually on products to control these internal parasites in livestock. And if you look at the animal health market in Ireland, products to control internal parasites are actually the single biggest component of the animal health market. All of the parasites that I mentioned previously are very prevalent on Irish farms. So some of them, like the gastrointestinal nematodes, would be ubiquitous. They'd be found on every farm in the country. Others, such as Fasciola, would be highly prevalent, although not present on every farm. And Fasciola itself is estimated to cost Irish farmers about 100 million euro a year in lost production. And it is particularly an issue in the north west or the western part of Ireland because of the weather conditions there, and particularly the wetter weather there, which favours Fasciola. Okay, so just to kind of briefly run through the life cycle of the liver fluke, Fasciola. So infected animals that have adult parasites in the bile ducts, those parasites lay eggs and they're passed out in the feces. If the weather's conditions are right, the eggs will hatch into myricidia, and this requires temperatures of above 10 degrees. These myricidia then have a fairly short period of time, a matter of hours, to find and infect a mud snail. And again, this mud snail replicates and becomes active at temperatures above 10 degrees. The parasite will then undergo replication within the mud snail before cercaria are released, and these cercaria then insist on the grass to form infective metacercaria that are ingested by grazing ruminants. They then pass through the intestinal wall and basically spend about maybe 10 to 12 weeks munching their way through the liver until they become adult fluke, where again, they start to lay eggs and complete the life cycle. Because there is a weather impact on the fluke life cycle, there is generally a seasonal pattern of fluke exposure on Irish farms. So a lot of animals in Ireland would be housed over the winter, housed for calving and lambing. And so there would be some Metacercaria that would survive over winter on the pasture, although these may start to decline throughout the spring. But if animals are turned out that are carrying fluke, these animals will then start to shed fluke eggs onto the pasture. And if the temperature and weather conditions are right, these fluke eggs will hatch and they will find and infect a snail, where the fluke will replicate within the snail, and then cercaria will start to be shed, usually later in the summer and into the autumn. So generally the highest risk grazing is in the latter part of the year, from autumn into winter, and this is when we see the majority of fluke infections. Because of this seasonal pattern, the Department of Agriculture, in collaboration with Met Éireann, can predict the risk of disease every year. And so every year they produce a liver fluke forecast. So they use rainfall and temperature conditions, particularly rainfall conditions throughout the summer period, to predict the risk of fluke in the autumn and the winter. And the fluke forecast for 2021 was just released about a week or so ago. And this here is a map of the risk of fluke infection throughout the country. So there are certain parts of the east of the country where there's quite a low risk of fluke this winter. Large parts of the country, I suppose there's predicted to be some fluke, and then particularly in the northwest, high risk of liver fluke. The Department of Agriculture also monitor lambs going through abattoirs. So they collect blood samples for serology and for the detection of antibodies against liver fluke. And they will map this throughout the year. So what you can see is you can see when the fluke infections start to appear in different parts of the country throughout the year. And again, this just shows the seroconversion survey from October 2021, where you can see in a large number of farms, the lambs have remained seronegative, but on some farms, particularly again in the northwest, seroconversion has occurred. So like all diseases, prevention is better than cure. So in terms of the steps that farmers can take to prevent fluke infection, things like draining localized wet areas to reduce snail habitat is one approach that they can take, although that obviously has implications for other organisms and besides the snail and might have kind of negative consequences for biodiversity. They can fence off the snail habitats, particularly in the high risk periods, or avoid grazing them in the high risk period. So if they have particular areas that are high risk, they may stop grazing them from, let's say, October on, or housing the animals again in the winter time will prevent them picking up some of this infection. And treatment with flucicides can also be used as a prevention measure. So if you make sure that animals are treated with an effective product, then the animals going out in the spring won't be shedding eggs and you'll be disrupting the life cycle in that way. In terms of diagnosis, an awful lot of farmers will know from their farm history whether they're at risk of fluke or not. In addition, the livers and lungs of all animals that go through abattoirs in Ireland are examined by vets. And in some cases, if they're signed up for certain schemes, these results will be returned to the farmer. So they may get these liver results that will tell them if the animal has a normal liver, if the liver is damaged but no live fluke were detected. So this might tell them the animals are at risk of fluke, but their treatment is working, or that the liver fluke is damaged and there is evidence of live fluke. And this can be particularly useful in terms of gauging risk for farmers. They can also use a fecal egg count for a diagnostic, but the one thing to bear in mind about the fecal egg count is the pre-patent period of fluke. So it takes about 10 to 12 weeks after infection before they mature to adulthood and start laying eggs. So they could be infected in that period, but they won't show up as positive on a fecal egg count test. There are also coproantigen tests that detect fluke proteins in the feces, and serum antibody tests that detect antibodies to fluke in serum in this case, I suppose, come with the caveat that while they're good at picking up infection early, those antibodies may hang around for a while after treatment, so they don't necessarily indicate an active infection. Blood enzyme profiles may give indications of liver damage, and what's important and was always recommended is, particularly in the autumn time for fluke, is that farmers would investigate sudden deaths. So if animals unexpectedly die and there's no obvious reason, that they're sent into one of the regional veterinary laboratories for a post-mortem in order to investigate whether acute fluke could be a problem. So in terms of control, in spring, the aim is to prevent the snails becoming infected by ensuring animals aren't shedding eggs. In the summer, to reduce the snail population. In autumn, to prevent animals grazing high-risk areas, and to prevent immature fluke migration and development. So this involves treating with a flukicide that is effective against immature fluke. And in winter, also treatment of at-risk animals to kill both adult and immature fluke to prevent liver damage and ill thrift. So a lot of this, as you can see, there's a fair bit of this involves the use of flukicides. And there's a large number of different products on the market, but they all fall into a reasonably small number of classes. And this first class here, triclobendazole, this is the only product that is effective against all stages of fluke. It's effective against early immature, immature, and adult fluke. Closantel and rifaxitinib are grouped together because these are somewhat similar. And so if a fluke develops resistance to one, it's likely they'll have side resistance to the other. So closantel, rifaxitinib, nitroxinol, they target immature and adult fluke, although nitroxinol is going off the market early next year. And then oxyclosanide and albendazole target only the adult fluke. So, there's not exactly a huge number of products that are available to farmers, and particularly in terms of products that target early immature or immature fluke, the choice is somewhat limited. And this is a real issue because of flukicide resistance, and anthelminthic resistance among all parasites is quite an issue for Ireland because of our grass-based system of production and because we're highly susceptible to these parasites and highly dependent on these drugs. Resistance to triclobendazole in Fasciola was first reported in 2009, and it's now believed to be quite widespread throughout the country. The other classes of flukicide are presumed to be effective, but that comes with the caveat that there's actually very limited testing going on for resistance. So there could be more resistance out there that we just don't know about. And as I mentioned, one class will cease production in 2022, and that leaves farms on which triclobendazole is not working. That leaves only one product available for the control of immature fluke on these farms. So finally, I suppose just to kind of reiterate that Irish agriculture is a really important Indigenous industry, and that internal parasites present a particular problem to us because of our grass-based production system. They negatively impact animal health and welfare. They've got a sustainability impact due to higher greenhouse gases and higher economic costs. Therefore, we really do need new and sustainable methods of internal parasite control. And there's a variety of different methods needed. So new management practices, also new diagnostics that would allow us to target our treatment better to infected animals or infected groups of animals. New vaccines would be a game changer by inducing immunity to fluke. And even if the vaccines weren't 100% effective, the fact that they would reduce fluke shedding means they could have a knock-on effect on pasture contamination. And so, even if they weren't 100% effective, vaccines would be really useful. And there's also a need for new anthelminthic products. So finally, I'd just like to thank all the people from the Parasitology and farm staff in Athenry and all of Teagasc, and our collaborators in NUI Galway in the Molecular Parasitology Lab, as well as other collaborators in UCD, the Department of Agriculture and Animal Health Ireland, and our funders, of course.
Lovely, thank you, Orla, for that very interesting talk. As I'd like just mention again, if any of the participants have any questions, we will be having a discussion at the end, not in between talks. So if you have any questions for Orla, please put them in the Q&A, not the chat. And with that, that'll bring us on nicely about anti-helminthic resistance and new practices to our next speaker, Krystyna Cwiklinski, where she's looking for novel ways or therapeutics in terms of vaccines now to combat this resistance. So Krystyna is a molecular parasitologist based at NUIG. She uses a combination of classical gene-targeted approaches and omic technologies to investigate drug resistance, parasite development, and host-parasite interaction to discover novel targets for vaccines and diagnostics. Currently, her work is focused on developing omics-inspired vaccines, the liver root parasite Fasciola hepatica, in collaboration with Teagasc, Cathepsin, Galway. So Krystyna is going to have her talk shown. It's pre-recorded, but we will show it now.
Today I'm going to present some of our work that we've been carrying out using an omics-inspired approach to identify targets for the development of a Fasciola hepatica vaccine. As Orla has already introduced, Fasciola hepatica, also known as the liver fluke, is an economically important pathogen of sheep and cattle, particularly in Ireland. Infection occurs following the ingestion of the infective metasecondary stage that exists as newly excisted juveniles or NEJs in the gut that migrate via the peritoneal cavity through the liver before moving into the bile ducts as sexually mature adults, producing thousands of eggs per day. Pathology associated with disease is caused by a variety of factors, including the damage caused by the migrating and feeding parasite, the host mechanisms used to repair this damage, as well as a range of parasite molecules that are secreted to aid its survival and migration through the host. Shown here are two pictures representing the acute stage of infection and the chronic stages of infection. And you can see here these white migratory tracts where the parasite has been moving through the liver tissue. In contrast, at the chronic stages, these migratory tracts have been resolved and repaired, although there is extensive fibrotic tissue indicating a heavy level of infection, as well as these engorged bile ducts. As the parasite is migrating through the mammalian host, it's encountering several different host tissues and microenvironments that it has to deal with. And at the same time, it's actively growing from the NEJ stage, that's approximately 0.1 millimeter in size, actively doubling in size every 2 weeks, resulting in the adult stage parasite that's about 2 centimeters in length. So to develop an effective vaccine, we have to have a good understanding of liver fluke biology to identify those molecules critical for parasite development and survival. The recent advances in sequencing technologies has really been a major step change advance for our understanding of liver fluke biology. Specifically because we now have the ability to investigate life cycle stages previously inaccessible to study, mainly due to their small size or the availability of sufficient material for in-depth analysis. So we now have the ability to investigate the development of the liver fluke parasite as it migrates through the mammalian host in a global and unbiased manner. The availability of the 2 draft genomes allows us to identify genes important for parasite biology and evolution and look at the gene structure, as well as identify elements critical for the regulation of these genes. Analysis of stage-specific transcriptome data from 7 developmental stages, specifically the metasecondary, the NEJ, the immature flukes 21 days post-infection, and the adult stage parasites, gives us information as to when and where these genes are being transcribed during parasite development. And from the same developmental stages, we have analysed the protein profile, identifying proteins crucial for metabolism within the somatic proteome, as well as identifying a range of molecules that are released and secreted within the excreted secreting protein fraction or secretome by the liver fluke parasite, which furthers our understanding of the types of molecules that are interacting and manipulating the host. Critical to our multi-omics approach to identify vaccine candidates for our liver fluke vaccine, and very much the starting point of our analysis, was the interrogation of the Fasciola hepatica genome. I mentioned on the previous slide that there are currently two versions of the Fasciola hepatica genome, and these represent a UK isolate and an American isolate, which allows us to investigate isolate or strain differences between geographically different liver fluke isolates, which may be important for our vaccine development and where our vaccine can be used globally. So the interrogation of the genome focused on identifying genes and specifically looking at their gene family structure within the genome. One thing that was particularly interesting and surprising was the size of the genome. So both of the Fasciola hepatica genomes are the same size, just over 1 gigabase in size. And this represents one of the largest pathogen genomes sequenced to date. Far larger than several of the closely related platyhelminths, and also far larger than those other worms that infect sheep and cattle. It is also about 1/3 of the size of the genomes of the mammalian hosts that the parasite infects. So we currently don't have an explanation for why the parasite genome is as large as it is. It could be seen as an evolutionary disadvantage to have such a large genome, particularly when the liver fluke parasites are producing all these thousands of eggs per day and the energy required to do so. However, our analysis has identified some key features that may in part explain why the parasite genome is as large as it is. One of these is that some of the genes that are known to be important for the parasite have expanded and duplicated into multi-gene membered families. Similarly, when we investigate the genomes of several different parasites from different geographical areas, we find that there's a huge propensity for variation across these genomes, also known as polymorphism. So today we're talking about Fasciola hepatica in the context of sheep and cattle infections. But Fasciola hepatica is known to infect a wide range of mammals, including those that it's most recently, evolutionarily speaking, has only just come into contact with, such as the kangaroo. And as Orla has also explained, one of the main problems in control of this parasite is that it has rapidly evolved drug resistance to the available drug classes used for its control. So the gene duplication and polymorphism that we see across the genome may be how the parasite has adapted to the range of mammalian hosts it infects, and also potentially gives the parasite the capacity for rapid evolution, which may be important in the context of the drug resistance that we're seeing. So on this slide here, I just wanted to highlight some key important genes for Fasciola hepatica development, which I will refer to later on in the talk. And those are the Cathepsin gene families, which encompass a range of Cathepsin L and Cathepsin B sequences. And these genes, proteins, are really important for the digestion of liver tissue as the parasite migrates, the digestion of hemoglobin during feeding, and also during immunomodulation by the cleavage of immunoglobulins. And what we can see here by this heat map is that they are differentially regulated across the life cycle. And by that I mean that different genes are expressed and transcribed by the different stages, indicating that they each play a distinct role at a distinct point in the life cycle. And this can also be said for the range of protease inhibitor gene families that we have identified, which also show a similar profile across the life cycle. This profile of expressing distinct subsets of genes and proteins throughout the life cycle occurs across the repertoire of liver fluke molecules being expressed, reflecting the very different parasite developmental stages within the mammalian hosts and the different niches that they are within and encounter. And really kind of represents the three major stages of the life cycle, that being of the invasive NEJ larvae that migrates across the intestinal wall, the migratory phase, this immature stage, and the final feeding mature stage of the adult parasites residing within the bile ducts. And as we can see from this heat map here, representing the different genes that are being switched on and off throughout the life cycle, we observed that there is tight regulation of these genes, but also that as the parasite grows and develops, it rapidly increases the number of genes that are being switched on. A similar profile can also be observed for the different proteins that are being secreted by the parasite, specifically shown here, the 3 stages, again representing those 3 major stages of the life cycle: the NEJs, the immature fluke at 21 days post-infection, and the adult parasite. And even within the proteins that they secrete, they are secreting distinct groups of proteins, indicating the specific role that they play in these 3 distinct niches. I spoke of the major advances that can now be made following the development and refinement of sequencing technology protocols, and in particular this relates to the fact that we can now analyse the parasite stages involved in early infection, namely the NEJs and immature fluke. If we are able to target these stages with our control strategies such as vaccines, it would minimise the extensive damage that the animal undergoes as the parasite migrates through the liver on the course during its development to the sexually mature adults. In particular, for this analysis, we focused on analysing the secreted proteins, as these are the molecules that are interacting and manipulating the host. We identified a range of proteins, including a number of those cathepsin peptidases that I spoke of on the previous slide, a number of protease inhibitors, antioxidants, and a range of immunomodulators that most likely play a critical role in manipulating the host immune responses to a more favourable Th2 wound healing environment. Our analysis of the profile of the proteins being secreted by these 2 stages indicate that the newly existed juveniles secretes a more varied profile with a large component being uncharacterised proteins, so of no known function to date. In contrast, the immature flukes at 21 days post-infection secrete a vast amount of proteases and protease inhibitors, in addition to a number of proteins that are involved in the digestion of blood, indicating that at this stage the parasite is developing into a more obligate blood feeder, rather than just feeding on the liver tissue. So from our proteomic analysis of the NEJ and immature fluke secretomes that I mentioned on the previous slide, we've selected a number of proteins as putative vaccine candidates, and these we're assessing in vaccine trials in sheep. We've used a combination of proteins in our vaccine cocktail rather than focusing on single proteins, and this is primarily due to the complexity of the life cycle and the developmental processes that the parasite's undergoing to really maximise the effect that our vaccine can have. We assess a range of parameters during our vaccine trials, including the level of parasite burden and a range of markers of animal health and productivity. So shown here are 2 trials that we carried out over 2 consecutive years, where we assessed the number of adult parasites recovered from the bile ducts at 16 weeks post-infection. At necropsy, we've assessed the number of eggs that are being released into the faeces that has the potential to contaminate future pastures. We've also assessed the viability of these eggs and in particular the impact that our vaccine can have on parasite transmission. And this was based on eggs recovered from the gallbladder, based on their ability to hatch under the right conditions. As I mentioned, we've also assessed a range of markers of animal health and productivity. And primarily this is due to the fact that typical clinical signs of fasciolosis, so liver fluke infection, include blood loss, anemia, and effects on growth rate and body condition, leading to reductions in animal productivity, subsequently leading to those economic losses that we've heard about. So shown here are the levels of hemoglobin, so at 16 weeks post-infection that could be associated with anemia, as well as the amount of weight gained by the animals during the vaccine trials. And the trial highlighted in blue, we saw some positive effects of our vaccine, indicating that although we may not have reduced the parasite burden, we may have impacted on the animal health and productivity, and allowed the animals to gain weight by vaccinating them with this cocktail. So just to briefly summarise, we have a large number of omic datasets available for Fasciola hepatica, and these can be used to investigate liver fluke biology on a global level that can also be exploited for the development of novel control methods and diagnostics. We've identified a number of key proteins that have shown significant impacts on parasite transmissions and animal health productivity, and we're currently trialling these in collaboration with Teagasc out in Irish pastures. So finally, I'd just like to thank the rest of the members of the Molecular Parasitology Lab, our collaborators at Morden Research Institute, and our collaborators at Teagasc, Orla Keane and Amanda McAvoy, and our funders SFI and ASC. Thank you very much for listening.
Okay, so that was the talk from Krystyna. So she will hopefully be available at the end of the talk for Q&A. So again, if you have any questions, please put them in the Q&A and we'll get to discussion at the end. So next we have Dr. Amber Dorey. So she has been working in the Molecular Parasitology Lab for the past 2 years as a postdoctoral researcher specializing in molecular biology. Her research focuses on the development of diagnostic techniques for detecting active liver fluke infections in sheep. The aim of her research is to develop a rapid point-of-care diagnostic in order to enable a more sustainable test then treat approach to dealing with infections and reducing anti-helminth resistance. So Amber.
Thank you very much, Richard. So I'm just going to share my screen with you. Okay. Okay, so today I'm going to talk, as Richie introduced me, on diagnostics for Fasciola hepatica, and I'm going to talk to you about both an enzyme-linked immunosorbent assay and also a more rapid lateral flow assay. So I don't want to go over too much about Fasciola hepatica because Orla and Krystyna have already done a great job of that. But I do want to just highlight a couple of things that I think are of importance. And first of all is the huge loss to the Irish farming industry that Fasciola infections cause every year, equal to about 100 million euros. And also how widespread the infection is, infecting around 50% of sheep flocks and up to 80% of dairy herds in Ireland. So it's huge. A major problem, which is why we need diagnostics, is the level of fluticide resistance in Ireland and also worldwide. And because of this, if we could introduce a testing then treat approach, this would enable us to do a targeted treatment. And also ideally, we want to try and develop a diagnostic which allows us to identify and treat pre-patent infections, so pre-egg shedding infections, to reduce pasture contamination. So there are currently some diagnostic assays available, and the gold standard of these is the fecal egg count. The fecal egg count, it can obviously detect eggs in feces, but it's quite a time-consuming and laborious assay to conduct. It can also be quite insensitive at low infection levels. And another problem with it is that it only detects actively shedding infections, so therefore infections that are effectively contaminating the pasture. So it's good, but it's not great. Recent approaches to diagnostic development have employed immunological techniques to try and improve sensitivity and also the accuracy of fluke detection. And one of these methods is the coproantigen ELISA. However, it's still, like the fecal egg count, insensitive at lower infectious loads. and it also cannot detect immature life cycle stages, only those which are egg shedding. More recent advances is the serological ELISA, which can detect fluke antibodies in blood samples. And this is what I want to discuss in more detail with you in a minute. So research in our laboratory has shown that as the liver fluke migrate through the tissues of the host, they release a variety of proteolytic enzymes during their infectious process. The cathepsin-like cysteine peptidases, they're produced by the gastrodermal cells of the parasite gut. And as Krystyna previously explained, they're strictly developmentally regulated. In adult flukes that are in the bile ducts, they produce the cathepsin L1 and cathepsin L2, while juvenile flukes in the intestines release cathepsin L3 and also the cathepsin Bs, so 1, 2, and 3. So research in our lab and also in other labs has shown that CL1 is highly immunogenic and therefore useful in serological detection of fasciolosis in both animals and humans. And in the research that I'm going to present to you now, the performance of all of these enzymes was compared and used to develop both the serological ELISA and also a rapid lateral flow test useful for diagnosing Fasciola hepatica infections. So the first step when producing the ELISA was to evaluate the efficacy of the 4 recombinant cathepsin peptidases, which we wanted to use for serodiagnosis of fasciolosis via an ELISA assay. So we collected serum samples from animals as experimentally infected animals at 0, 3, and 16 weeks post-infection. And we compared the level of the antibodies to the 4 cathepsins at 3 and 16 weeks post-infection compared to 0. So at 3 weeks post-infection, we could see a clear increase in the level of antibodies against these cathepsins compared to our 0 weeks. But the highest level that we detected was in our CL1 and CL3. The level of all 3 of the CLs increased drastically at 16 weeks post-infection compared to 3 weeks. However, the cathepsin B, it stayed stable. There was no change in the levels between 3 and 16 weeks post-infection. So we also wanted then to evaluate how the antibodies changed at 3 weeks post-infection compared to 0 weeks, because we're trying to develop a diagnostic to detect pre-patent infections. So when we compared the antibody levels 3 weeks post-infection and 0 post-infection, we could see that for CL1 and CL3, we got a significant increase in the antibody levels at 3 weeks compared to the pre-infection serum. While there was an increase for the CL2 and also the cathepsin B, this wasn't significant. And overall, we saw that for the CL1, we could detect significant increase in 91% of infections, and for the CL3, 95%. And while in the CL2 and the cathepsin B, this was reduced to 40 and 41% respectively. So the current goal, or the current treatment for fasciola is triclobendazole. So we wanted then to test whether our ELISA assay would be able to assess seropositivity both pre and post treatment with triclobendazole. So to do this, 11 animals were experimentally infected with liver fluke. And at 17 weeks post-infection, 6 of these animals were administered a dose of triclobendazole, which was calculated based on their body weight. And we wanted to monitor their antibody levels. So in our control group, well, in both groups, but focusing first on the control, we can see that from 0 weeks all the way up to 17 weeks, we get a really good increase in antibody levels. And in our control group, this kind of plateaus around the 15-week mark and stays fairly constant thereafter. The exception to this was the cathepsin B, which peaked between 7 and 11 weeks post-infection and then started to decline, so it's not great long-term. When we then look at our treatment group, so these received their triclopentazole at 17 weeks, we can see that the levels of the CL1, 2, and 3, it sharply declines post-treatment. So this suggests that these antibodies would be good for assessing antibody levels post-treatment as well. So because they decline, we might be able to use these ELISAs to pick up new infections. So animals that are infected, treated, the treatment is effective, but then become reinfected from the pasture. So that's our ELISA assay, and the ELISA, it's very good levels of sensitivity and specificity, but there are some drawbacks with it, and the fact that it can be quite time consuming and laborious to perform, and usually you're waiting at least kind of a day for your results. It also requires some expensive equipment such as a plate reader. Lateral flow assays, they offer comparable levels of sensitivity and specificity to the ELISA, but the pros of these is that you can have a result within 30 minutes. Probably the most famous of the lateral flow assays available is the home pregnancy test, which can provide a result within 5 minutes using an easily obtained urine sample. So they're simple and user-friendly, and little to no equipment is required other than the lateral flow test itself. These have also become more popular during the COVID pandemic as the rapid antigen tests. So we wanted then to see, we have this great ELISA, can we now do further development and produce a lateral flow test? So in order to do this, we collected serosamples from 5 animals that were experimentally treated. So we collected serosamples at 0, 3, 7, and 14 weeks post-infection. And when we tested these serosamples by ELISA, 2 animals, so animals C and D, were positive on the ELISA at 3 weeks post-infection, and all 5 animals were positive at 7 weeks post-infection mark by ELISA. When we also ran them on the lateral flow assay, 1 animal, so animal D, which had quite a high antibody response, we could detect that they were infected at as little as 3 weeks post-infection. And like the ELISA, all animals were positive on the lateral flow assay at 7 and 14 weeks post-infection, suggesting that the lateral flow assay offers similar kind of levels of sensitivity and specificity as the ELISA assay does. So to conclude, when we looked at all of the Cathepsin Ls and also the Cathepsin B, we found that the CL1 and 3 perform better compared to the CL2 when we were looking at the diagnosis of acute fasciolosis. The levels of antibodies against all 3 Cathepsin Ls remained high during chronic infection, but they all do decline after treatment. The Cathepsin B levels, they start to decline around 7 weeks post-infection, suggesting that this antigen may only be useful as a marker for acute infection, whereas the Cathepsin L is good for acute and chronic infection. The ELISA assay is a useful diagnostic for detecting subclinical infection, which is particularly good when we're trying to diagnose pre-patent infections and treat them prior to the shedding of eggs in the feces. The lateral flow assay, it demonstrates similar levels of sensitivity and specificity as the ELISA does, but the benefit is that you can get a result within 30 minutes as opposed to having to wait a day or so for your result, and you don't need any expensive equipment to do it. Both the ELISA and the lateral flow assay are able to detect subclinical infections, allowing targeted treatment prior to egg shedding and feces, but they also provide a huge leap compared to the existing fecal egg count and the copper antigen ELISA. And finally, it's important to acknowledge everyone that's been really key in this research. So everyone in the molecular parasitology lab, but also the molecular biotechnology laboratory at Maynooth University, as the lateral flow aspect of this project has been carried out as part of a collaboration in this lab with Professor Sean Doyle, and also a help of Orla and Amanda in Chagas collecting samples and running the trials, and finally to SFI for their funding. So thank you very much.
Lovely, thank you, Amber, for that. That was great. Now, again, if you have any questions, please direct them in the Q&A, not the chat. And it would be helpful if you actually say who the question is for as well. So next we have Nicola Calvani. She is a member of the Molecular Parasitology Lab. She completed her PhD in molecular diagnostics and differentiation of Fasciola species in Southeast Asia at the University of Sydney. During this time, she developed a suite of diagnostic tools to differentiate Fasciola hepatica, Fasciola gigantica, and mixed hybrid infections from fecal samples of ruminants, which she then used to screen local cattle and important livestock trading corridors in northern Laos. I think we're going to hear now the conclusion of that. Go ahead. Up to you.
Thanks.
Over to you, Eva.
Let me just share my screen. All right, is that visible? Okay, so hi everyone, and as Richard said, my name's Nicola Calvani, and I'm here today to talk to you about Fasciola outside of Ireland, and specifically by that I mean the interplay between Fasciola hepatica and its sister species, Fasciola gigantica, in Southeast Asia. And before I get started, I obviously need to thank Orla, Krystyna and Amber for doing such a fantastic job introducing Fasciola hepatica and for sparing me from spending time going through the life cycle treatment and diagnosis. So I'll get more into the specifics about Fasciola gigantica in a second, but first I wanted to point out that like Fasciola hepatica, Fasciola gigantica is not only a significant parasite of animals, but it's also a significant human pathogen as well. Globally, between the two of them, up to 91 million people are considered at risk of infection, and women and children are more likely to be infected. Recent estimates using an existing economic costing that has been updated for inflation suggests that at a prevalence of about 60%, the production impacts of Fasciola gigantica in Southeast Asia alone could be as high as $1.8 trillion US. And obviously that's quite extreme, but given the ubiquity of this parasite in the region, it's not surprising that the cost of infection on production could be so high. Fasciola hepatica is primarily found in temperate countries such as here in Ireland and in Australia and the UK, but Fasciola gigantica, on the other hand, is usually found in tropical areas, including throughout Southeast Asia. The geographic distribution of these parasites has traditionally been associated with post-domestication livestock movements, as you can see in this map here. Given the historical distributions of these parasites, it's perhaps not surprising that the ever-growing international trade of livestock is likely to be quietly changing the status quo of their known distributions due to the increased movement of animals from Fasciola hepatica endemic to non-endemic regions. In Southeast Asia in particular, the combination of aquatic rice production with large ruminant husbandry provides an ideal scenario for both human and animal fasciolosis. Here, a growing middle class has seen an associated increase in demand for animal-derived protein. And as a result of this increased demand, over 150,000 animals per month were imported into China in 2019 alone, of which 70% were from Fasciola hepatica endemic countries such as Australia, Argentina, and Brazil. Now, while the conditions supporting Fasciola hepatica translocation events might be most apparent in Southeast Asia, it's important to note that similar Fasciola species movements are likely to be occurring elsewhere, such as in the Middle East, where sheep and goats from Fasciola hepatica endemic countries are routinely imported into the region. Now, I thought I'd pause for a moment and indicate what livestock production actually looks like in Southeast Asia, because it's very different to what it looks like here in Ireland. And although it's still considered a highly seasonal parasite, much like it is here, as was pointed out earlier by Orla, the seasonality of infection in tropical countries is largely driven by the fluctuation between quite distinct wet and dry seasons. So here you can see that aquatic rice production is the predominant agricultural output, and it provides the perfect environment for the intermediate snail hosts to thrive in. Rice production is either low input and entirely rainfall fed, or it can be high input aquatic irrigated production where multiple crops are produced each year. These farming systems will influence the seasonality of infection along with climatic conditions, where animals become infected after grazing rice stubble after harvest, or when they're fed rice straw as forage. So now you might be thinking, if Fasciola gigantica is already present in Southeast Asia, as you can see in the map here in yellow, do we really need to be concerned about the introduction of Fasciola hepatica? Traditionally, between the two species, Fasciola hepatica has been considered sort of the worst one due to increased reports of zoonotic cases, higher parasite burden in infected animals, and an assumed greater pathogenicity than that of Fasciola gigantica alone. These assumptions could be true, or they could simply reflect sort of increased scientific scrutiny in developed countries where Fasciola hepatica is predominantly found and where access to medical care and treatment options is more widely available. Importantly, however, in areas where Fasciola hepatica is common, there are widespread reports of anthelmintic resistance, as was pointed out by Orla. Resistance to any drug class has yet to be reported for Fasciola gigantica, but given that there are so few available with high efficacy against both the adult and immature forms, it's imperative that we try and limit the spread of drug resistance in both species at all costs. And it's especially concerning in areas where the human fasciolosis is common, such as in Southeast Asia, because it's the only drug that's registered for use in people. Now, as you can see here in blue, the distribution of Fasciola hepatica in Southeast Asia is limited for now, but alarmingly, you can see here in green that there is a wider occurrence of Fasciola hybrids, which suggests that perhaps Fasciola hepatica has yet to be detected in many of these countries. So in the graph at the bottom here, you can see that there have been an increasing number of reports of hybrids between Fasciola hepatica and Fasciola gigantica in the scientific literature. And this is primarily occurring in regions of known parasite symmetry, such as throughout Southeast Asia. Despite records of these Fasciola hybrids continuing to increase, we still don't have any information about the functional implications of these forms. So we still don't know, what do they actually mean for human and animal health, and do we need to be concerned about drug resistance emerging in them? Experimentally, successful hybridization between the two species has been demonstrated in the lab, and the resulting F1 and F2 generations have displayed like an intermediate morphological phenotype between that of the two parental species. These lab-generated hybrids appear to be more infectious than Fasciola gigantica alone, based on higher than previously reported recovery rates in Fasciola gigantica in experimentally infected animals, but there has been limited work on this. Importantly, hybridization doesn't necessarily generate a permanent genetic change. And so these intermediate phenotypes could be inconsequential in the long term, especially if you consider the limited viability of the successive generations in lab experiments. But while the hybrids might only exist transiently, in the grand scheme of things, a more permanent and therefore concerning consequence of these interspecific mating events are the outcomes when animals are co-infected and back crosses between hybrids and either of the original parental species can occur. And that's because it allows potential for introgression of advantageous traits from one species into the other. So introgression is the transfer of genetic material from one species to another. And as I said, it usually occurs as a result of repeated back crossing of hybrids with either of the original parental species. The occurrence of introgression between Fasciola species is as of yet unproven, but I've theoretically represented it here in this diagram. But most importantly, adaptive introgression is the establishment of beneficial alleles in a population, and it could occur as a result of various forms of selection pressure. So here I've got a simplified diagram which demonstrates the potential random drift of introgressed alleles from Fasciola hepatica in blue into the genome of Fasciola gigantica in yellow, and the fixation of new alleles at a single locus. As you can see, initially many alleles introgress, but the incompatible ones are selected against and will eventually disappear. However, by generation 500 down the bottom there, the advantageous allele is moving towards fixation and the others persist at lower rates of recombination. The source of selection pressure is population dependent, but it could involve adaptations for drug resistance, increased virulence, adaptation to new definitive or intermediate hosts, higher metasaccharide output, increased temperature tolerance, the list goes on. Adaptive introgression has already seen the transfer of resistance genes against organophosphates and pyrethrum between Anopheles mosquito species, as well as the experimental induction of ivermectin resistance in lab-maintained Homunculus contortus populations. And so that's another common parasite of livestock, Barber's poleworm. So you can see it's not altogether unprecedented or even inconceivable that adaptive introgression could result in a transfer of anthelmintic resistance in Fasciola species due to the importation of triclobendazole-resistant Fasciola hepatica into Fasciola gigantica endemic regions where appropriate selection pressure is applied. Introgression between these two species, if it exists, could have other important human and animal health impacts as well, and that could include increased infectivity, virulence and pathophysiology, not to mention any potential implications surrounding infection of intermediate hosts or survival of eggs and the infectious metasaccariae in the environment. So I've just said that we don't know if these events are happening in these parasites. So why not? And how do we go about finding if they are? Well, differentiation between hybridization and introgression is not easy in these parasites. Firstly, because of their hermaphroditic nature, they have an assumed potential for parthenogenesis, and there's the occurrence of polyploids as well, which collectively these things complicate our understanding of these events and our ability to detect them. But the major ambiguity that surrounds the distinction of hybridization from introgression in these parasites is due to a tendency for people to investigate single loci to confirm species identity when a multi-loci or even a whole genome approach is required. So when I put together the species distribution map of Southeast Asia that you saw a couple of slides ago, I was really careful to only include cases where each Fasciola specimen had been molecularly confirmed in local animals and using a minimum of 2 genetic markers. And I've tried to explain why here in this diagram. So unless a marker has previously been identified as being susceptible to introgression in these species, single markers are not appropriate for differentiation. So as you can see here in both of the pure specimens on the left and even the hybrid in the middle there, accurate identification can theoretically be conducted regardless of the locus targeted in a pure population. But in a mixed population or an unknown population and in introgress specimens, as you can see here on the right, the markers selected could misrepresent the true status of that specimen. So that's been shown here in ticks, where the marker selected has appropriately identified the specimen, but across where an additional marker has suggested that inappropriate identification has occurred. Further, I've also shown in other work that traditional sequencing technologies, such as Sanger sequencing, can even itself misrepresent the true identity of known hybrid forms due to preferential amplification of dominant DNA. But ultimately, it's quickly becoming clear that without knowing more about the specific genetic makeup of these forms, the overinterpretation of naturally occurring hybridization and introgression events should really be avoided, and identification should be conducted with care and also with a thorough understanding of the intricacies involved and the limitations of existing technology. The availability of the draft Fasciola hepatica and gigantica genomes, as mentioned by Krystyna, as well as increasingly accessible next generation sequencing technologies will hopefully enable documentation of introgression in these parasites going forwards. Just to quickly conclude, the continued neglect of these parasites and their human and animal health impacts in the developing world mean that very few studies have actually examined the role that the international trade of livestock plays in the expansion of existing hybrid zones. but the little evidence that we have available is damning. And when it's considered in relation to what we know about the impacts of introgression in other parasite species, it really does beg the question, is introgression between Fasciola hepatica and Fasciola gigantica just a fluke, or is it something more? Both co-infection and hybridization have been proven in Fasciola and are likely to be occurring with increased frequency due to the movement of Fasciola hepatica infected animals into Fasciola gigantica endemic regions. And I hope that I've shown in my brief presentation today that while there's no evidence in support of adaptive introgression between these parasites just yet, it's simply a matter of when and not necessarily if before we start detecting them. So it's therefore time that we actually start considering as a scientific community the longevity, functional and epidemiological implications of these events in order to determine the impacts that they could play on food security and public health. And if you want to know more about hybridization and introgression in these species, then scan this QR code and read my opinion on the matter in Trends in Parasitology. And also just a quick note that an updated chapter of Fasciola gigantica in Southeast Asia will very soon be available in the second edition of John Dalton's essential reading on this topic, the fasciolosis textbook. So I just need to say, obviously, a big thank you to all of you for listening, but also to my fantastic lab mates shown here, and many of whom who have presented today, for helping me learn so much more about the biology of this parasite since coming to Ireland a year ago. Thanks.
Lovely, thank you very much, Nicola. And I'd like to thank our 4 panelists for their great talks. And now I'd like to hand over to Caroline Verissimo, who's going to be moderating the Q&A and discussion afterwards. Thank you. Carolina.
Thank you, Richard. So first, thank you to Dr. Orla Keane, Krystyna Cwiklinski, Amber Dorey, and Nicola Calvani for their excellent presentations. On my opinion, they gave a very good overview about the liver fluke problem to agriculture in Ireland and also in other parts of the world, as Nicola highlighted there. So I have a few questions here. I will try to make it even between you all guys. So firstly, to Dr. Orla Keane, can you hear there, Orla?
Yes.
Yes. So Devla Morris asks you, are there actions being taken to combat anti-helmintic resistance, and how do these relate to actions outlined in INEP2?
Okay, yeah. So I suppose, you know, being probably fairly blunt about it, we're probably a little bit behind where the antimicrobial resistance field are. So there is a national action plan to deal with anti-parasitic resistance. Mm-hmm. However, it only commenced in 2020. So we're a little bit behind the INAP plan. Now, it very much kind of follows the structure of the INAP plan. So it has the same kind of overarching strategic objectives in terms of improving awareness and knowledge of antiparasitic resistance, enhancing surveillance of antiparasitic resistance and usage, reducing the spread of disease, optimizing the use of antiparasitic medicines, and promoting research. So it has the same broad strategic objectives. But it only launched last year. There are about 40 actions. I think there are 42 or something actions in the plan. So it's in train, but we're behind where the antimicrobial resistance field would be.
Okay. Thank you, Orla. Another question is there any program for eradication of parasites like the federal government have for other diseases like FMD, brucellosis? Quite similar, maybe you know.
Yeah, so at the moment there aren't, and I suspect that there probably won't be. So in terms of national eradication schemes, they would be very, very carefully evaluated in terms of what disease would be likely, and that there would be a likely success and that they would be able to eradicate. Obviously, they've had success eradicating brucellosis. They have a ongoing program at the moment to eradicate BVD. However, the parasitic diseases are much more complicated. So in terms of, there's a very large reservoir of them in the free living stages. Some of them also have, as well as the intermediate hosts, they might have wildlife hosts or that. So I think the chances of success with an eradication program are extremely, they're just not there. And I mean, any attempt to even eradicate parasites locally on a kind of an on-farm level has never been very successful because there's a really large reservoir outside of the host. So no, there are no plans to do that.
That's difficult, not only for Fasciola, but for other parasitic diseases as well. It's the management for to eradicate a parasitic disease is very hard. So
yeah.
Why we would like to focus in vaccines as well. I guess that's the best strategy, in order to eradicate any or try to eradicate any parasitic disease. Thank you, Orla. I have other questions for you, but I will try to not keep only the focus on you. So let's change to Krystyna Cwiklinski. Can you hear me, Krystyna, there?
Yes, I can.
So, Krystyna, a question here is, clearly Fasciola hepatica falls in that category of a difficult to target pathogen, where we need to understand very well the protective immunity that of this hosts to develop a good vaccine. Do you think we are in this point we understand enough about the protective immunity or the immunity that the hosts develop when naturally infected by Fasciola?
To a short answer, no, I don't think we fully understand how infection plays out within the animals. Obviously, we know that the parasite secretes a range of molecules that we term immunomodulatory. And by that, we mean obviously that they actually interact and manipulate the host immune response. And we're only really starting to get a handle on what some of these molecules might be doing, but in a more kind of lab-based environment. So we need to kind of learn more about how these act in vivo. And also, as Orla pointed out, more often than not, these animals are infected with more than one parasite. So, we can look at how Fasciola affects the animal, but equally we need to understand what the other parasites, what the other infections, both parasite, virus, bacterial, also have an effect on the immune response when we think about our vaccines and the immune responses that we want to actually induce.
Thank you, Krystyna. Another question is, if a vaccine, like mentioned during the talks, would work, like not necessarily has to absolutely prevent the infection, but could be very effective just reducing the pathogenicity and therefore preventing all the economical losses related to the infection. Therefore, there is this, I saw on your presentation that mainly you focus on looking for molecules that are differentially expressed by different life stages of Fasciola hepatica. Do you think we should be targeting an antigen that is co-expressed throughout or expressed throughout the whole life cycle to have like more chances that the antibody response would work and maybe damaging the parasite at different stages to reduce this pathogenicity observed?
So yeah, that's one approach that we could take by looking at something that's expressed throughout the life cycle that's very dominant and we can target at all stages. Another avenue that we're actually looking at is that unlike, say, the COVID vaccines that we've all been receiving, we're using a vaccine that's targeted against multiple proteins. So we can actually then target these different stages by actually using a combination of different antigens in our cocktail. And this is primarily important because obviously, as we've seen in the life cycle, the parasite rapidly moves, it develops, it changes, it's not staying the same throughout that kind of period of about 10 weeks of infection before it reaches the adult stages. So yeah, I think we definitely need to be targeting the different stages so that we can maximize the effect that we're having. But there's different approaches that we can take to maximize that.
Thank you very much, Krystyna. Really, really good insight on this. I'll change a little bit again. Let's ask Amber. Are you there?
Yeah, I'm here, Carol.
So Amber, Seamus Fagan asks, what matrix is used in the lateral flow test? Serum?
Yeah, so at the moment we've been developing it using sera samples, the same as the ELISA, but the technology is available to run ELISA assay, lateral flow assays with whole blood, which would obviously be easier for a point of care diagnostic. There's also the potential to use other fluids, for example, milk. If we were to test cattle, that might be an easier sample, because if you have milk, you don't then need a vet to come out to take a blood sample. That would allow point of care testing, but obviously there's some issues with that as well, which we'd need to consider carefully beforehand. But the technology would be there to expand it from sera to include other bodily fluid samples.
Okay. Thank you, Amber. Just highlighting here that if any person on our audience would like to ask questions, just raise the hand and feel free also to ask alive here. Amber, just another question here for you is, do you plan that these lateral flow tests will become available on farmers for in situ diagnostics? And if yes, do you think that this is ideal, like having in my mind like the possible over-treatment and the fluke-side resistance? Like, what's your view on this?
So, there would be, I suppose, pros if it would become available to farmers, especially, for example, if they could use it for monitoring like bulk milk tanks to monitor the herd in that way. But then we do have the problem in that you may get over treatment. For example, we see when we treat with triclobendazole, even if the treatment is successful, it takes several weeks for the antibody response to come down. So you could run into a problem where you have someone who treats and then tests, say, a week later, and they still see a positive. So they keep treating when they don't really have a positive response. Now, I suppose because there's going to be new legislation coming in from January 2022, where you will need a prescription to purchase anti-parasitic drugs. So, I mean, maybe with that brought in, there could be more opportunity, because obviously, instead of just getting your positive lateral flow test and running off and buying an antihelmintic, you would need a prescription. So that would allow some degree of sort of vet input to prevent someone continuously treating every week or whatever, because they keep getting a positive result. So there are pros to that, but there's also cons. And I think it would need to be very carefully looked at. And also there would need to be proper training available in terms of courses, seminars, etc., to provide good levels of education about how to use the tests correctly and how to also interpret the results with them.
Yeah, that's very important. I guess the combination of this legislation with the test is ideal. Otherwise, like I would think that the best is getting this test available to the vets and allow them to use this and to have like a fast diagnostics and very sensitive. Thanks, Amber. For now, I have a few questions to Nicola Calvani. If you are there, Nicola, please.
Yep. Hi, Carolina.
Hi, Nicola. So, Nicola, is there any method available to identify hybrids or introgression on material, for example, fecal samples containing eggs, fossil eggs?
Carolina, how nice of you to ask that question. So I spent the majority of my PhD trying to devise a diagnostic method to distinguish species from fecal samples, because much like on farms here in Southeast Asia, it's quite difficult to collect blood samples, and even if you could, the ELISAs unfortunately don't differentiate between the species present, and it's almost impossible to get access to the adult flukes, which is what species identification is traditionally done based on, because it's hard to get access to abattoirs in Southeast Asia. It's quite a politically sensitive topic. So in that respect, I spent a long time developing qPCR methods that would detect DNA from eggs in fecal samples. And then if you used a selection of markers, you could say that you had either a Fasciola hepatica infection, a Fasciola gigantica infection, or a sample that had mixed DNA. But the limitation of that was because you're looking at DNA from many individuals in a sample, you can't distinguish if that mixed DNA is because you have a hybrid present or if because you have co-infected animals present. And then, of course, the caveat there is that we still don't have markers for introgression. So regardless of what marker you used, you couldn't say that this animal had introgressed forms inside of it unless you were able to collect the adult specimens and then had an appropriate marker to test with. But that is one other thing, is that we do now have at least samples that allow screening from minimally invasive samples of species that are present. And it's one of the ways that we confirmed that Fasciola hepatica was present in northern Laos.
That's very good. Really, congratulations for developing the method. And the other question is there any regulation that China puts in place, for example, to import animals, to assess the animals imported from the endemic countries like Brazil, Argentina, Australia, before importing, or even after receiving the animals there? Are these animals being assessed or tests or quarantined? Is there any regulation that you know of?
Yeah, that's a really great question. And it's also, again, quite a politically loaded question. So countries do get quite touchy about the importation or export of live animals and the screening of diseases they need prior to export. And currently, no countries sort of require screening for Fasciola, either species. But also one thing that's important to note is that once animals do arrive in Southeast Asia, the movement of animals through the region and predominantly up into China, because that's where many of the animals are going, is largely undocumented. So there's a lot of transboundary animal movement that is through unofficial channels. And so that is how we actually anticipate that Fasciola hepatica ended up in the region, because there are leakages from these systems. Animals sort of disappear out and end up on local farms or in local systems there. So although there may be requirements for testing on arrival, because there is such an extensive sort of journey for them through Southeast Asia, and that system is largely unregulated, it's quite difficult to keep a close eye on it and to monitor it. One thing that I will point out is that when we were screening northern Laos, which is a largely widely known livestock corridor into China, Laos has a border with China, we found that a concentration of animals that we detected Fasciola hepatica in were found in a region that had a large Australian Angus beef cattle farm in the region. So our theory is that animals potentially came over from elsewhere in the world and were infected and then shed into the local environment, or there was sort of mixing of grazing areas, and that's one way that they could have become infected. But at the same time, it's very difficult to point that out explicitly, but as I said, there's been documentation at least of animals that have arrived in abattoirs in the Middle East that have been co-infected or infected with Fasciola hepatica. So it's certainly happening, but it's difficult to keep tabs on it.
So other difficulties of controlling parasitic diseases, this movement, I guess, more and more like in the modern world. There is another question here, Nicola, is when you are working there and with the animals in Asia, could you note a difference in terms of the health of the animals infected with Fasciola gigantica, Fasciola hepatica, or even the mixed infection that you saw there, like in some animals, or it's hard to notice this differences. The question is, because we are so used to Fasciola hepatica, I think it's important for us to understand a little bit also.
Yeah, no, thanks. That's a really great question. Unfortunately, because at the time, this was pre-COVID that I did all this work. So at the time, there was very limited molecular diagnostic capacity in Laos. So I collected these samples over a period of 5 months, but then wasn't able to actually process them until I arrived back in Australia. So I wasn't able to collect the sample and then go back and view the animal and sort of determine if there was any change. But regardless of that, it would have been difficult to differentiate sort of production impact anyway, because animals in Southeast Asia, in Laos and Cambodia, often are chronically malnourished because of the fact that their predominant diet is either roadside grasses or dry rice straw from the rice harvest, which has very low protein content. So these animals typically look to have depressed production outcomes. So that's one aspect. And then you also have to consider that there are several other quite important production limiting diseases that occur in Laos, including sort of foot and mouth disease, hemorrhagic septicemia, etc., as well as other parasites. So it's very difficult to rule out a differentiation between, production impacts of the various things that are affecting livestock production there. But that's certainly an area that I would like to look into more, is what are the impacts on animal health, but also human health, differences between Fasciola hepatica and Fasciola gigantica infection.
Thank you, Nicola. For now, I will come back to you soon, hopefully. So, Amber, there is a comment, actually not actually a question, but it's just adding to what you were talking about, the new regulations coming next year. So Amanda Lalor, I hope that I am pronouncing your name properly, Amanda, says that the prescription rules have apparently changed from January to June 1st next year. So ICOS confirmed this information yesterday after the AFM meeting. So just for us to know, which is basically a pity, would be nice having this implemented from the beginning of the year, I guess.
Yeah, I think I think putting them prescription only is good in terms of antihelmintic resistance and helping to avoid that. But I understand there's also a lot of implications in terms of cost for farmers involved now with having to get a prescription. So I think possibly delaying the implementation of that while allows more time for talks to determine what else needs to be done, so that it's not farmers suffering necessarily with the increased cost implications of this.
Thank you. Also, Nicola, an anonymous person here, cannot see the name, just would like to tell you that he thanks a lot for your presentation, and hope that you working in the future in other countries, including Pakistan as well. Here.
Well, thank you. And I should also point out that, yes, although my presentation focused on Southeast Asia, it's certainly a problem in countries in East Asia and in the Middle East, such as Pakistan and Iran as well. So thanks.
Okay. Orla, you mentioned during your presentation that old animals develop a certain resistance to parasitic infections. Do you understand or do we have any understanding on how this resistance happens, or any mechanisms behind this, or not really? Because, of course, we don't observe this with Fasciola. I guess there's no correlation between age. It's just more common for us to observe infection and treat in lambs because we want to prevent this, like these animals to grow or to have the impact of the infection. But I was just curious.
Yeah, so I suppose, for a number of the parasites, immunity does develop. One thing to point out is that it's rarely sterile immunity, so that the animals they can withstand the impacts. They will have lower numbers of parasites, but they won't have sterile immunity. The mechanism is really going to depend on the various parasite species, and even varies between, animals or between breeds. And obviously there's an awful lot more that we could know about that, so in some of the work that we've done looking at the impact of the gastrointestinal nematodes in sheep, what you find is that in some breeds, immunity, for want of a better word, manifests as fewer worms in the gut, whereas in other breeds, immunity manifests itself as shorter worms that produce fewer eggs, because generally we're assessing this by assessing the fecal egg count. So we're using an indirect measure of resistance to these diseases. So it doesn't necessarily always mean the same thing between different animals and different breeds. And there's also variation between animals in terms of their levels of resistance to these diseases and how well immunity develops. And that's something that is exploited or can be exploited in terms of breeding animals and selecting those animals that have superior resistance to the disease.
Thank you very much. A last just question to Amber before we finalize the session. Actually to Amber. It's actually related to what we were talking with Orla Keane there. Did you assess on your tests the interference of co-infections on animals? If there is any impact on this test that you are developing, ELISAs or lateral flow?
Yep, so certainly for the lateral flow, we have looked to see if co-infection of animals with rumen fluke has any effect, and we have found nothing. And we also know that animals that are infected with rumen fluke but not with liver fluke are negative, so we don't seem to have any cross-reaction between the two. We've also looked at sera from animals negative for liver fluke but infected with a variety of nematode species, and we found no cross-reactivity there. So far, to the best of our knowledge, our test is highly specific just for liver fluke.
Thank you, Amber. And with this, I will move to finish this session. Again, I would like to thank you very much to all our amazing speakers in this session, and for their high quality presentations, also for answering the questions here. I also would like to thank the audience for their participation and for their questions that, of course, improve our discussion. Let's thank you very much also to the communications team and to the Ryan Institute and to the Center for One Health for helping us to organize this event and to make it happen so smoothly. The same to Siobhan Galgan and Professor John Dalton that put a lot of time and effort for the session happens. So if you would like to engage with us in our research group, you can find us on Twitter, Facebook, and Instagram. You can search for Molecular Parasitology Laboratory, or MPL, at NUIG. And in there, you'll find more information. We are always sharing information on our research and other things related to liver fluke and other parasitic diseases. Thank you very much for all, and see you later.